Assalamualaikum and salam sejahtera. We are from group 18. Today we are going to present on topic 6, which is non-replicating viral vector and DNA vaccine. So our group consists of 4 members. My name is Siharda binti Cik Hassan and my other 3 members are Nurhanani Nadia binti Muhammad Yusof, Auni Fasiha binti Zamri and Nur Dianisha Ashrani binti Muhammad Rudi. We're going to start our presentation with introduction of non-replicating viral vector and DNA vaccine.
Before we go through, we have to know what is the introduction of non-replicating viral vector or DNA vaccine. As we all know, non-replicating vaccines are based on recombinant viral vector that are made replication non-competent, meaning that these vectors are sufficient to induce host immune response but cannot replicate inside host cell. In addition, RNA-based vaccines that involve mRNA sequence for coding pathogen-specific protein, or also known as antigen inside host cell, that can be generated using non-replicating mRNA. Non-replicating viral vector or DNA vaccine. Non-replicating viral vector have several viruses, which are adenovirus, adeno-associated virus, measles virus, and human parainfluenza virus. Viral vectors that are modified to make replication defective that are called as non-replicating vector. Eventually, the virus gain an attenuated state. It can still be able to trigger the human immune response but cannot replicate in human cells. Adenovirus vaccines are capable of inducing both antibody-mediated and T-cell-mediated immune response. However, the intensity of response depends on the virus serotype used to produce the vaccine. Principle of how the vaccine works. The immune system, which also known as the body defense against infection, is the one of the principal vaccine work. To understand how COVID-19 vaccine work, it helps to first look at how our bodies fight illness. Our immune system uses several tools to fight infection. Blood contains red cells, which carry oxygen to tissue and organ, and white or immune cells, which fight infection. There are different types of white blood cells fight infection in different ways. First, macrophages are white blood cells that swallow up and digest germs and dead or dying cells. The macrophages leave behind part of the invading germ called antigen. The body identifies antigen as dangerous and stimulates antibodies to attack them. Second, B lymphocytes are defensive white blood cells. They produce antibodies that attack the pieces of the virus left behind the macrophages. Next, T lymphocytes are another type of the defensive white blood cell. They attack cells in the body that have already been infected.
So now let's move on to the example of vaccine developers. For AstraZeneca vaccine, it is developed by pharmaceutical industry company. This one, it is a British-Swedish multinational pharmaceutical and biotechnology company where the headquarters are at the Cambridge Biomedical Campus in England. For the Sputnik V vaccine, it is developed by Gamalaya Research Institute of Epidemiology and Microbiology. Next, for the Sputnik V vaccine efficacy, the vaccine showed around 97.6% efficacy. This based on mass vaccination in Russia, where 3.8 million Russians were received Sputnik V from 5 December 2020 until 31st March of 2021. This based on disease infection rate among people receiving both shot of the Sputnik V vaccine. The infection rate was around 0.027% starting from the 35th day of the first dose, and the disease occurrence among unvaccinated adults was 1.1% from the 35th day after the mass vaccination in the Russia. For the AstraZeneca vaccine, it is reported that the vaccine has efficacy of 76% after the first dose, where the protection is still maintained until the second dose. So after 12 weeks and more, its efficacy increased to 82%. These data were based on the primary analysis of the phase 3 clinical trials from the UK, South Africa, and also Brazil. No severe cases and no hospitalizations were reported after more than 22 days of the first dose. Also, the analysis showed that the potential for the vaccines to reduce asymptomatic transmission of the COVID-19 viruses. This was based on the weekly swabs obtained from volunteers in the UK trial. PCR positive readings were also reduced by 67% after a single dose and 50% after getting 2 doses. So as you can see, based on this Google image, um, they compare COVID-19 vaccines of AstraZeneca and Sputnik V. So just look at the effectiveness of the vaccines. For the AstraZeneca, it is 62 until 90%, while for the Sputnik V, it showed 92%.
Next, the vaccine trial conducted. The vaccine trial conducted involved two steps of testing, which is animal testing and human testing. For animal testing, animals are given different doses of vaccine to check for adverse events and an immune response. There was no animal model for COVID-19 because the virus that caused it is too new. According to the new research, ferrets, cats, and certain non-human primates have all been found to be susceptible to infection and can transmit it to others. Information about safety and perhaps efficacy was provided by animal testing before a vaccine is tested on humans. Next, for human testing, it can be divided into three phases, which is phase 1, phase 2, and phase 3 vaccine trials. Let's see to the phase 1 vaccine trials. This phase involved 10 to 100 healthy volunteers to receive the different doses of vaccine. Blood is taken from the volunteer to measure the immunological response to the vaccine. Specifically, the risk in antibodies and whether antibody levels are similar to those seen in patients who have had the illness. For the phase 2 vaccine trials, this phase includes hundreds of volunteers from a wider range of backgrounds, which will provide a better indication of the vaccine safety and immunological response among people who are likely to receive it in the future. Due to the limited number of participants involved in phase 1 and phase 2, they only give information on vaccine side effects that are very common. So the only way to assess vaccine efficacy is to measure antibody levels. And lastly, for the phase 3 vaccine trials, this phase involves tens of thousands of volunteers who receive the vaccine can give crucial evidence that vaccination is both effective and safe. In these trials, participants are given either the vaccine or a placebo. We have direct evidence that the vaccination prevents disease when fewer persons who receive it get sick than those who receive a placebo.
Advantages. The main advantage of DNA vaccines is their ability to stimulate both the humoral and cellular arms of the adaptive immune system. In regards to humoral immunity, the generation of antibodies by B lymphocytes against invading pathogens is one of the most effective defense mounted by the immune system. DNA vaccines are efficient at generating T-cell response that may kill targeted cancerous cells or cells that are infected by the target virus or bacteria. Next, it is also used as a therapeutic to treat chronic infectious diseases such as HIV and hepatitis C virus. DNA vaccines are easy to transport and store. Next, DNA vaccine also stable molecule and does not need to be stored at low temperatures, making transportation and storage cheaper and easier than conventional vaccine. Lastly, DNA vaccines are easier to make large amounts of a gene than make proteins or grow up bacteria or viruses. Next, the disadvantages of DNA vaccines. DNA vaccine can disrupt normal cellular process that change foreign DNA into the body could affect a cell's normal protein expression pathway. Next, DNA vaccine also can make chronic inflammation because the vaccine continuously stimulates the immune system to produce antibodies and recognize the agent as foreign, destroy it, and remember it. In addition, DNA vaccines can make initial attempts to create DNA vaccines have not worked because they have not had a big enough impact on the immune system. Lastly, no DNA vaccine has been licensed for use in humans yet. Although some DNA vaccines are now in clinical trials, none are licensed for use, so they are an unproven method.
Last but not least, side effect to human or post-vaccinated effect. As we all know, after getting vaccinated, we may experience some mild side effects which are signs that our body is building protection. This is because our immune system tells our body to respond in certain ways, such as increasing blood flow to allow more immune cells to circulate and raising our body temperature to destroy the virus. The majority of COVID-19 vaccine side effects have been mild to moderate and have lasted no longer than a few days. Typical side effects include pain at the injection site, fever, fatigue, headache, body ache, muscle pain, chills, diarrhea, and nausea.
In conclusion, not all vaccines to prevent COVID-19 will be successful. However, non-replicating vaccines, DNA vaccines, are very safe as there is a very low risk of vaccine antigen-induced disease onset. That's all from us. Thank you.